Learn about DNA and their components. As you can see here, this is a DNA model. You can see that every human being, every living creature has DNA strands copies in each and one of them. Okay? Now, what is DNA? DNA stands for deoxyribonucleic acid. It is a self-replicating material which is present in nearly all organisms. Okay, so we're going to dive into that. We're just going to do this part 1 today, and in the next video we'll dive into part 2. Okay, so let's dive in. Now, to start off, I'm going to start off with these two guys named James Watson and Francis Crick. These are the guys that pretty much understood and discovered the structure of the DNA, how the DNA is, that is a double helix. And think of it as two ladders, but twisted. So for order for DNA to fit into every cell in our bodies, it has to be coiled up a lot of times, multiple times, so that it could be packaged properly and fit into your cells. And thanks to the DNA transfers information to the next generation. And it replicates itself so that it keeps that pattern, genes, processes that we benefit from and that we use on a daily basis. Now, here's some components to understand here. The structure of a nucleic acid. So here we have nitrogen bases, which is, when you see here, the green. So these are adenine, guanine, cytosine, thymine, and uracil. These are bases that are used in the DNA. Okay. Uracil only occurs in the RNA, just for correction. Adenine, guanine, cytosine, and thymine occurs in the DNA. Okay, RNA has the rest, but except thymine. So instead of having thymine, they replace it with uracil. Now, sugars, the blue here, can be deoxyribose in the DNA or ribose in RNA. Okay, and phosphate has a negative charge, which is the backbone of the DNA. Okay. Here we have nitrogenous bases. So let me move this up a bit. Good. So DNA has no uracil. RNA has no thymine. Okay. So here in the top we have pyrimidines, meaning they're hexagonal shape, meaning they have 6 sides, and that's thymine and cytosine. Also uracil and cytosine. Well, cytosine, of course, is the same here. Also pyrimidines. So basically there's 3 types: thymine, cytosine, and uracil. Okay. Now here in the bottom we have purines. They have a hexagonal shape combined with a pentagon shape. So what does that mean? Purines, A combines in DNA, A combines with T. Adenine matches up with T, and cytosine matches up with G, okay, or guanine. And RNA, since there's no thymine, adenine has to combine with uracil, okay, and cytosine still combines with guanine, okay. So here's an example how that looks. You see adenine here, this sugar combines normally with thymine. It only has 2 hydrogen bonds. Hydrogen bonds are weaker, so it's easier for them to open it and close it. Here, guanine has 3 bonds. Hydrogen bonds as well, but it's more than A to bonded to T. So GC has 3, AT has 2. So those are purines. So the purines are adenine, A, guanine, G, and the pyrimidines are thymine, T, cytosine, C, and the pairing is this one. A only has 2 bonds to T, 2 bonds, and C has 3 bonds, as you can see here. 2 bonds from A to T, and 3 bonds from C to G. Okay? And here's the DNA double helix structure of DNA. This is how this looks like when you a 3D image in color, double helix style, this is how you look at it in 2D, partial chemical structures with the bonds, you can see it. And here's a space filling model, a 3D physical model that you can actually see. Okay, so you can see how all of that is looking like and formed together. But they're all the same. Okay? So that is just a basic recap of what a DNA helix structure is. Its function is it provides the blueprint for information to make different cells and different tissues and everything in our body. And when you mate, you pass down that DNA through your gametes. Okay? This is the first part of DNA replication. I'll see you in the next video where we're going to talk about part 2. Okay? This is Mr. Frederick, keeping it real and keeping it simple every day.